Okay, everyone. So in this video, what we're going to be looking at is the actual individual muscle fiber. Okay, so we talked about what the skeletal muscular system does, right? Its main functions. We talked about the broad structure of the organ itself, right? But let's get to our most basic unit that's what's inside of the skeletal muscle, which is the actual muscle fiber. Okay, so we're going to be specifically looking at the muscle fiber that is a skeletal muscle fiber. The fibers that you find in cardiovascular muscle. In cardiac muscle and find in smooth muscle are structurally a little bit different. We're mostly interested in what's happening again with our skeletal muscle in this particular case. Okay, so there are a few terms that we're going to start by defining just to understand the basic structure of it. Okay, so the first is that when we're talking about a muscle fiber, again, a muscle cell, there are a few different parts to it. So like with any cell, there's going to be a cell membrane that surrounds it. So specifically when we're talking about skeletal muscle, the membrane is known as the sarcolemma. Okay, so the sarcolemma is the cell membrane of the muscle fiber. The reason why we call it the sarcolemma is that anything sarco just refers to, again, muscle, specifically the muscle fiber. Okay, but this unique name is just because structurally the membrane is a little bit different. There's some different pathways that different ions and things can move through that aren't necessarily present in other cell structures. But the cell membrane, we just call it the sarcolemma. Okay, so within the muscle cell, we have a different name for the inside of it. So we call it the cytoplasm in normal cells, or in cells that aren't muscle cells. But in muscle cells, we call it the sarcoplasma. Okay, so the sarcoplasm in this particular cell is going to be different in composition compared to what you get in other types of cells. Okay. The other things that you're going to see that are unique to muscle fibers is they're going to have these structures called T-tubules, which are transverse tubules. And these are essentially openings that tunnel into the sarcolemma. And it's mainly important for increasing surface area within the muscle cell. The reason why we increase the surface area here is to allow for the transmission of particular ions that help aid in the actual contraction of the muscle. Okay, and we'll get to that a little bit later. But what you need to know is that structurally the cell is going to be behaving a little bit differently. Okay, it's going to be like all of these indentations that happen inside of the cell just to increase surface area. Okay, all right, so we got sarcolemma, sarcoplasm, right, and then these T-tubules that increase the surface area. Okay, some other terms that we need to know in this particular example are the myofibrils. Okay, so myofibrils are, again, related to our muscle fiber. They're 1 to 2 micrometers in diameter and length of the muscle fiber. So myofibrils are just kind of like filaments that are inside of the muscle fiber. Okay, and we'll talk about them in a little bit more detail here in a second. Within the myofibrils, again, these like links of muscle fiber, you're going to have some different filaments that function differently. Okay, so you have myofilaments. Myofilaments are made up of thick and thin proteins. Thick and thin proteins, again, that's just like a broad general term, because when you're looking at it under a microscope, for example, you can kind of see these, especially if you're at like really, really high magnification with a really, really good scope. But really with these myofilaments, you need to know the names of the individual pieces. So there's actin, which is functioning as the thin filament, and myosin, which is the thick filament. Okay, so myosin, that myo, again, prefix, root means muscle. So again, these are thick filaments that are found specifically in muscle. Actin is found in a lot of other places inside of the muscle cell and inside of any cell in general. Okay, but these thick and thin muscle proteins inside of the cells allow for the cells to actually do muscle contraction. Okay, and we'll talk about the kind of steps of muscle contraction in a little bit. Okay, right. So some other things to think about in terms of our muscle fiber that makes it unique, right, is that with our muscle fiber, it's going to have a heck of a lot of mitochondria. So as you guys all probably remember, right, the mitochondria is responsible for the breakdown of sugars and turning those into ATP. So it's like the powerhouse of the cell. The idea is that they are responsible for metabolizing a lot of different materials necessary for your muscles to do the contraction. Okay, so it's responsible specifically for the breakdown of glycogen. So glycogen is actually a complex carbohydrate. Again, a lot of glucose molecules put together in a specific way that we store energy inside of our muscles. We use it to store energy inside of our muscles because we need that rapid available resource of sugars in order for muscle contractions to occur. Okay. Your muscles generally store a high amount of glycogen. It takes anywhere from 3 minutes to 15 minutes, depending upon the particular muscle type, for you to actually utilize all of the glycogen. And you have more glycogen stored in other parts of your body that you can rapidly send to your muscles to allow you to continue to do your muscle contractions. This is why you have to work out for a certain amount of time, is to get rid of that excess source, so you start using other sources of energy like fat inside your body. Okay. But essentially, it's just a quick, easy source for your muscles to get to in order to generate enough ATP for either muscle contractions that you have to do a lot of them, so a lot of reps, or where you require a lot of extra force or energy in order to make those pushes and pulls, right? It's like, say, if you're lifting a bunch of stuff or carrying something really heavy. Okay. Other structures that are inside of your muscle cell that are kind of unique. So the first one here is the sarcoplasmic reticulum. The sarcoplasmic reticulum is unique in our muscle cell compared to what we saw in just the general cell type. Right? We know that the smooth muscle is responsible for packaging up different materials. Here, the sarcoplasmic reticulum is actually a smooth ER that's specialized within the muscle fiber to help transmit or move high concentrations of calcium ions, which are responsible for contraction. Okay, so these are directly linked to those T-tubules, again, those areas where you have the sarcolemma going down into the muscle fiber to increase the surface area. The smooth endoplasmic reticulum or sarcoplasmic reticulum in your muscle cells directly linked to that. And what they do is they store high concentrations of calcium ions, which are needed for that muscle contraction, specifically releasing it in areas that we call the terminal cisternae. Okay, so a cistern is like just kind of like a area where you store things, like sort of like I just think of it like a big pot. But the portable cisternae is just kind of an area where there's a high exchange of ions across membranes. Okay, so structurally, the skeletal muscle fiber has these unique structures here. Right? Again, features that are unique to a muscle fiber as a whole. Okay, and there are some other things that we'll look at in terms of images regarding the muscle here in a little bit. Okay, but within the muscle, the big structure that we need to go over and make sure that we understand is the actual structure that provides us the ability to contract. So this is actually the part of your cell that is actually able to shorten the length of the cell, right, in order to allow you to pull on the bones in order to get motion. Okay, right, and that structure is known as the sarcomere. Okay. So the sarcomere, when we're talking about, again, functional units inside of your muscle cell, the sarcomere is the contractile unit inside of your myofibril, okay? So again, it's that part that's actually inside of your muscle tissue that allows you to do the contraction that's actually doing the pulling, okay? So again, this is a microscopic structure that's within a myofibril, right? There are millions of these within muscle tissue, right? And they all work together to help shorten the muscle fibers, which then shorten the whole muscle, right? Because if you shorten a muscle fiber, the fascicle shortens, everything shortens, and you're allowed to pull on the muscle or on the bone of interest. Your sarcomere is responsible for doing that, okay? So again, per myofibril, so just like per muscle cell, or per part of muscle cell even, you can have as many as 10,000. You can actually end up with more than those depending upon what muscle you're talking about and where you're talking about it and how fit and how large your muscles are. And these are arranged in a particular order. They're kind of ranged and stacked kind of on top of each other, which ends up when you're looking at the muscle tissue, you can see this kind of striation pattern, which allows you to kind of see where the parts where there's certain structural proteins that kind of gives you this banding that you see. And you can see that under a microscope really well. It's harder to see on a more macroscopic scale, but essentially this striation is what allows us to see muscle tissue separate from other things. When we talk about muscle tissue as a whole, like skeletal muscle is also called striated muscle, right? The idea that you're able to see these particular structures inside of the cell forming these banding patterns. So seeing sarcomeres doing that, okay? So let's talk about the structure of the sarcomere. So here I have an image of what the sarcomere looks like. So if you want to pause for a second so you can sketch this out, right? What essentially you're seeing here is the basic functional unit inside of your sarcomere. Okay, I'm going to go through and define what each of the individual parts are within this. But again, I encourage you to just kind of pause, draw this thing out. I would recommend drawing it in 2 different colors just so you can see the difference between these fibers here and these fibers here. Again, they structurally are different. They're different proteins. Just so you know what's there, because I'm going to move down and talk about each of the individual parts as we look. Okay, all right. So let's talk about each of the individual pieces that are embedded within this image. So I'll see if I can zoom out just a tiny bit. so we can kind of see all of it. Okay, actually that's bad. Let's zoom in a little bit just so we can talk about the individual parts. So I'll kind of go back and forth. All right, so the first boundary, so when we're talking about the actual structure here, we go from a particular area that we call the Z line. So the Z line is, you can see that over here, and call it the Z line because it forms this kind of zigzag pattern back and forth, sort of like the letter Z. And this is the boundary point between each individual sarcomere. So we go from Z line to Z line, and that is one sarcomere. Okay. Within this, you're going to have a couple other areas. Okay. So you can see the M line. So the M line is this band of protein that's right down the center. The protein at the center that connects the thick filaments together. So these blue ones are the thick filaments, and the thin filaments are these red ones. Okay. You have a couple other bands that you can see structurally happening inside of the muscle tissue. Okay. So the first is the A band. Okay. So the A band actually goes across the from one end of the thick filament all the way to the end to the other. So this is forming our A band. Again, it's kind of this area that's thicker in the part. When you're looking at the striation, it's kind of the darker part of the striation. And then the lighter part is what you're going to see around the Z lines. Okay, so the I band is the thin filament end to the end of the A band to the next A band. Okay, so that's just structurally to give you an idea of what's there. So if we go from the end of this thick filament across the Z line to the other end of the next thick filament, that is the I band. So again, generally this is the area that's kind of see-through in the striation. It's a little more see-through in coloring because you just have less stuff compared to everything here. All right, and then lastly you have the H band. So this is thin filament to thin filament across the M line. Okay, so you're looking at this part here to this part here. Okay, so structurally those pieces there, the Z line, M line, A band, I band, and H band, those are relevant and important to us to understand the structure of the sarcomere because these are the functional things that are actually moving when you have the contraction of the muscle fiber. Okay, so in the video I'll talk about the specifics of what's going on with that muscle contraction, but essentially just to give you a quick rundown of what that means, is that if we take these muscle fibers, right, essentially what they're going to be doing is like pulling on the bands and making them move. Okay, so it's like by pulling on these things, the muscle fiber gets closer and closer together. The H band starts to shrink, right? And then when the muscle relaxes, the H band gets larger. Okay, so that's kind of our goal here, is to kind of shrink the H band, right, and then decrease, and then increase the H band as we're doing our contraction. Okay, so that's what's going to end up happening with our sarcomeres. Okay, so I'll dive into more detail about the specifics of muscle contraction, all the parts that are there. Right, so if you've got more questions, let me know.